Bismillah ar-Rahman ar-Rahim. Today we will describe the muscular tissue data show. In this picture we can see the longitudinal section of the skeletal muscle. Also we can identify certain labels like this dark line in the middle of a lightly stained area is the Z line. And this light area on both sides of the Z line is the light band or the I band. And this dark area here is the A band or the dark band. Also, this is the nucleus. And these are the skeletal muscle fibers. As regards the questions that may be asked in this picture, you may be asked about the size of the skeletal muscle fibers. It is from 10 to 100 micrometer in diameter. You can be asked about the shape. What is the shape? The shape is, of course, cylindrical, not branching, except in few areas in the body. Also, you may be asked about the staining of the cytoplasm. It is acidophilic. Or the picture of the nucleus. The nucleus is oval, multiple, and peripheral in position. Suggest why the nucleus or the nuclei here are multiple. Why they are multiple in this skeletal muscle fiber? They are multiple because the fiber is formed by fusion of mononuclear myoblast. Mononuclear myoblast. So by this fusion we have multiple nuclei. Of course, you may be asked more in depth. Mention one organelle which is present in these fibers. The most prominent organelle here is the myofibrils. And also we have another organelle not present in the picture, which is the mitochondria. Also mention one inclusion. You can mention either glycogen or myoglobin. You may be asked why the Z line appears, or what is the composition of the Z line. You may be asked about the composition of the Z line. Of course, you will answer that the Z line is formed of actin. Here, the Z line is formed of actin together with the matrix, together with the other proteins. This is the Z line. What is the composition of the A band, which is the dark band? It is formed of both actin and myosin. What is the composition of the I or the light band? It is formed only of actin. This picture shows also longitudinal section in the skeletal muscle fiber. Here we have a label for the nucleus. We have here multiple peripheral nuclei. Also we have a label for these striations, which are transverse striations. Also we have a label for the connective tissue, very delicate connective tissue in between the cylindrical muscle fibers. As regards the endomysium, you may have a question about the composition of this endomysium. This endomysium is formed of reticular fibers that surround each muscle fiber. And in this endomysium, small blood vessels and small nerves are present. So this is as regards the endomysium. As regards the striations, you may be asked what is the cause of these transverse striations. The cause is that the presence of dark band in the myofilaments on the same level and light bands in the myofilaments also in the same level, giving the appearance of transverse striations for the skeletal muscle fiber. This picture is a transverse section in the skeletal muscle fibers. Why it is transverse? Because the muscle fibers are not cylindrical, but they are mostly polyhedral or polygonal or rounded in shape. Why it is a skeletal? Because we have peripheral nuclei, and in some of the sections they appear to be multiple nuclei. Then you have in this slide we have a very important thing, which is the connective tissue material in the skeletal muscle. First we have this very thick connective tissue, which is the epimysium. This epimysium covers the whole muscle, and you should remember it is formed of dense connective tissue, and it is containing this dense connective tissue. It contains large blood vessels and large veins, large nerves. Then from the undersurface of this epimysium we have more fine connective tissue, or more or less dense connective tissue coming from the undersurface surrounding the bundles of the skeletal muscle fibers. So we have this one, which is less thick and less dense, which is the perimysium. It is formed of less dense connective tissue and contains also blood vessels and nerves, but of smaller size than the peripheral one. And finally you will find this very delicate connective tissue in between the muscle fibers. This is the endomysium, and it is formed of reticular fibers containing very fine blood vessels and the nerves. So the very thick on the surface is the epimysium, formed of dense connective tissue. Then the connective tissue surrounding the bundle is called the perimysium, and it is formed of less dense connective tissue. And inside the bundle itself, around the individual muscle fibers, we have a reticular connective tissue, which is the endomysium. Again, this is a picture for a transverse section. Why transverse? Because we have the polygonal transversely cut skeletal muscle fibers, and the nucleus is peripheral in position, and some of the fibers demonstrate multiple nuclei. Then you may be asked about the connective tissue which surrounds this bundle here, this one. This one is the perimysium, formed of less dense connective tissue and containing blood vessels and nerves. And then we have the very fine reticular connective tissue in between individual muscle fibers surrounding them. This is the endomysium, containing fine blood vessels and nerves. Here we have a blood vessel here, which is present in the perimysium, and the epimysium. You cannot judge where is the epimysium because I cannot see the whole surface of the muscle. So we have perimysium and endomysium. In this picture we also have a transverse section in the skeletal muscle fibers, the same previous questions, and these are the nuclei peripherally located, but here we have additional informations, which are the presence of these single cell layered blood vessels, which are minute capillaries here present in the endomysium because they are surrounding the individual muscle fibers. Also we have two different types of skeletal muscle fibers. We have this one which is dark and one which is lightly stained. So this is the red fiber and this is the white fiber. You may be asked here, what are the differences between the red and the white? The red fibers are characterized by being rich in three things. What are these three things? They are rich in myoglobin. Here we have the red fibers are rich in the myoglobin, and also rich in the blood vessels or the vasculature, and also they are rich in the mitochondria. So by this way they are responsible for the aerobic respiration, because the myoglobin responsible for carrying the oxygen, and the blood vessels for the supply of the oxygen, and the mitochondria for the energy, so they are responsible for aerobic respiration. On the other side, they are poor relatively in the glycogen, which is the material used in anaerobic respiration. So the red fibers here are characterized by these characters, by being rich in the blood vessels, the myoglobin, the mitochondria, and poor in the glycogen. On the other side, we have the white fibers. In case of the white fibers, which here can be seen, which is less stained than the red, this is the red and this is the white. The white fibers are contrary to the red because they are poor in the myoglobin, poor in blood supply, poor in mitochondria, and they are responsible for anaerobic respiration, and accordingly they have a big store of glycogen which can be used in this type of respiration. This picture is for the cardiac muscle here, and this prolongation which is called the valve. So it is a cardiac muscle and valve. In case of this prolongation, you have outer covering and inner covering. Here it is called the endocardium. It covers the whole surface of the valve and line the ventricle and the auricle or the atrium. In this valve, here we have this red area. It is called fibrous connective tissue. This fibrous connective tissue may be asked, what is the composition of this fibrous connective tissue? It is formed of 3 elements. Number 1, collagen fibers. Number 1, the collagen fibers to give support. Number 2, the elastic fibers to give stretch and recoil. Number 3, we have the macrophages. Macrophages to phagocytose any antigen or foreign material. In this area, the extension of this fibrous tissue here, this extension covers the cardiac muscle. So in this area, it is called supporting tissue. So this is the supporting tissue, this is supporting tissue, and this is the fibrous tissue. This is the endocardium, this is the endocardium. All of this is the endocardium. All of this is the endocardium. This material is the cardiac muscle fibers cut in different directions. Some are longitudinal, some are oblique, some are transverse. In this picture, you can diagnose that it is a transverse section in a muscle. Here, the fibers are not cylindrical, but more or less polygonal or rounded in shape, so it is a transverse section. Then, it is a transverse section in the cardiac muscle. Why? Because we have a central rounded nucleus. Here we have a central rounded nucleus. Almost all of the transversely cut fibers have a central rounded nucleus. So it is a cardiac muscle fiber. You may be asked also about this tissue containing RBCs. This is the endomysium in between the individual muscle fibers. Also, you may be asked about some theoretical knowledge, like what is the size of the cardiac muscle fiber. Here the size is different from that in the skeletal. It is 15 micrometer in diameter. Also about the shape of the nucleus, it is single, central, and rounded nucleus. Also, as regards the organelles, we have the myofibrils here, organelles, and the mitochondria. These are the organelles. As regards the inclusions, we have glycogen and we have the lipofuscin, which is accumulated material that increase with the advance of age, causing brown atrophy of the heart. So these are the possible theoretical questions in this slide. The size of the cardiac muscle fiber 15 micrometer, and the prominent organelles, myofibrils and mitochondria, and inclusions, glycogen and lipofuscin, and the appearance of the nuclei. The nuclei are central, rounded, single nucleus. Of course, in this section, the nuclei appear rounded because actually it is a transverse section in an oval nucleus. So the nucleus actually here it is oval, but this in the longitudinal section. If you cut it transverse section, you will see it rounded in shape. So the appearance of the nuclei in the transverse section is rounded, while in the longitudinal section is oval in shape. This picture shows the longitudinal section in the cardiac muscle. Mention the features that help you to diagnose that it is a longitudinal section. First of all, the fibers here appear to be cylindrical, so it is a longitudinal section. Why it is cardiac muscle fiber? Because we have the nuclei here. This is the nucleus, which is oval, and it is more or less achromatic or pale with prominent nucleolus, and it is present in the center of the segment. Take care that these dark nuclei are not those of the cardiac muscle, they are of the endomysium, in between the cardiac muscle fibers. This is the first feature, the appearance of a single central oval relatively pale nuclei. The second feature is the appearance of a darkly stained lines, these which are called intercalated disc. So these are the features that help me to diagnose that it is a cardiac muscle fiber and it is a longitudinal section. Another question may be about the function of the intercalated disc. The intercalated disc has two functions because it has two parts. It has a part formed of desmosome, so this will prevent the separation of the segments from each other, and it has another part which is in the form of a gap junction, and this will allow the spread of the impulse from one segment to another very rapidly to follow the all or none rule. Also in this picture you may diagnose that we have two nuclei, and this always occurs at the site of branching. This is a branching of the cardiac muscle. So what are the criteria of the cardiac muscle here? The branching, the single central oval nuclei, the intercalated disc. This is another picture for the Purkinje fibers. These Purkinje fibers are actually modified fibers, have certain characters that are very specific to them. You have to study what are the characters of the Purkinje muscle fibers, which are different from the ordinary cardiac muscle fibers described before. First of all, the first character is that these fibers here, the fibers here are large. This is the first specific character. Second character, the nucleus here is not central as the ordinary cardiac muscle, but it is here, as you will see here, it is peripheral or eccentric in position. It is eccentric in position and may be binucleated also. So this is as regards the nucleus. Then we have the sarcoplasm. Here the sarcoplasm is rich in glycogen that appear in the form of vacuoles. So it is a vacuolated cytoplasm. Here you can see, here we have the vacuoles inside these fibers. We cannot see these vacuoles in the ordinary cardiac muscle fibers. Actually here also we have a very specific character, which is the presence of very few myofibrils. Few myofibrils. And actually these myofibrils most probably have a specific arrangement. They are very few at the periphery, and in the center they almost always take the Mercedes mark, or they take this picture also. We have this is the peripheral, and then we have this arrangement of the myofibrils in the center. So you may see this picture of the arrangement of central myofibrils in the form of Mercedes mark or in the form of this cross. So these are the characters of the Purkinje fibers. You may be asked about the function. You may be asked what is the function of these Purkinje fibers. The function is that they spread the impulse, cardiac impulse, very rapidly compared to the cardiac muscle fibers, ordinary cardiac muscle fibers. This is as regards the function of the Purkinje. Another picture for the Purkinje fibers you can see here. This is the Purkinje muscle fibers. You can see the centrally located myofibrils. You can see the eccentric nucleus. You can see the vacuolation of the sarcoplasm. So this is the Purkinje muscle fibers, much magnified. Move to the third type of the muscle fibers, which is the smooth muscle fiber. The smooth muscle fibers are not cylindrical. They are in the form of spindles. The spindle means that it is a structure which is wide in the center and narrow at the periphery. And they have single central oval nuclei. Here is the nucleus. This is the nucleus present in the center of these spindle-shaped muscle fibers. Sometimes they describe the nucleus looking like a cigar, so it is called cigar-shaped nucleus. And this blue tissue, it is the connective tissue stained here in between the muscle fibers. So it is collagen or connective tissue containing collagen fibers but stained with a specific stain here. This picture you can see also the longitudinally cut smooth muscle fiber spindle, typically spindle in shape, wide in the center, narrow at the periphery, with a single central oval or cigar-shaped nucleus. And we have empty spaces of endomysium in between. Here we have a different picture for the smooth muscle. It is transversely cut. And of course, when this spindle is cut in the periphery, you will have a small circle. When it is cut in the center, you will have a much bigger circle, and you will notice also some of them the nucleus. When we have in the other side, which is also small, you will have a small section. So if you look to this picture, you have some of the sections have a nucleus inside, so it is most probably the central portion, and we have other portions which are devoid of the nucleus, and this of course the peripheral parts that do not contain the nucleus inside. And we have this white area, it is a connective tissue, very fine connective tissue in between. Here a picture for a longitudinal section in the smooth muscle, and because we have both longitudinal and transverse, so that in the exam you should specify either the section is longitudinal like this or transverse. So this is a longitudinal section. Why? Because the fibers appear as a spindle in shape. And the nucleus here is single oval nucleus. So once it is oval in shape, so it is also a longitudinal section. This is the soft connective tissue or the fine connective tissue in between the muscle fibers groups, and of course containing blood vessels. This is a connective tissue. And if you look to the muscle fiber itself, as we said, it is an spindle in shape, acidophilic cytoplasm, and the nucleus is of the active type mostly. They have pale nucleus with oval shape, and they are single central in position. Also in this section you should remember the size. We have discussed the shape, but about the size we have the following data. As regards the size, you should remember the numbers 8 and 20 and 500. The number 8, it is 8 micrometer, which is the diameter of the smooth muscle fiber. So this is the diameter. The number 20 micrometer and the number 500 micrometer, actually it is the range for the length of the smooth muscle fibers. 20 micrometer, it is in the wall of the blood vessels, while the 500 micrometer, it is the length of the smooth muscle fibers in the pregnant uterus. Pregnant uterus, of course, is much enlarged, so the length of the smooth muscle fibers reaches 500 micrometer, while in the blood vessel it is 20 micrometer in length. Now let's start with this trial exam. In this picture, of course, it is evident that it is a transverse section in the muscle. Here in the section we have the blue arrow points to this connective tissue on the surface of the muscle. So it is the epimysium. Epi means above, mysium means muscle. And then we have this broad, relatively connective tissue surrounding the bundle. It is the perimysium. So the green arrow points to the perimysium. In the other section on this picture, we have, of course, transverse section in a muscle. You have to identify, is it cardiac or skeletal? If you look here, you will see the nucleus in a peripheral position. So this is a transverse section in the skeletal muscle, and here you can see two nuclei in this transverse section. So it is a skeletal muscle fibers. Also we have an arrow, this blue arrow pointing to the nucleus. And we have an arrowhead which points to the blood vessel having a lumen and formed of one layer of flat cells, flat squamous cells, so it is a capillary. Another one here but containing few RBCs, and another one here and another one there, so these are capillaries. Of course, the capillaries are present in the three layers, epimysium, perimysium, and endomysium, but their size decreases with entering to the inside of the bundle. Here also we have this is, of course, is a blood capillary, and we have a lot of blood capillaries in the connective tissue and the muscle fibers here. If you look to this one, you can see it is dotted. These dots represent the myofibrils. Also, it is a picture for the skeletal muscle, and you should mention the direction. The direction here, it is a longitudinal section. Why? Because the fibers are cylindrical in shape. Why it is skeletal? It is cylindrical, non-branching, and it has the multiple peripheral oval nuclei. So it is an LS in the skeletal muscle, and we have these the nuclei. But what about these nuclei in the connective tissue in between? Look at this nucleus, it is very dark, and this one it is pale. So these dark nuclei are the nuclei of the endomysium, and the pale nuclei here, relatively pale, it is the nucleus of the skeletal muscle fiber. Here another picture, and by the first look it is a longitudinal section because I can see cylinders. Then next step, to find out what type of the muscle. We have here a central nuclei. This is very evident, central nucleus, and we have this line which is the intercalated disc, and we have branching of the fibers, so it is the LS in the cardiac muscle. And this line, as we said, it is the intercalated disc. It is formed of two types of junctions, desmosomes and gap junctions, and we have mentioned the function of each one. Here another picture which demonstrates a group of fibers which are paler than the surrounding fibers. This group of fibers paler and appears to be rich of glycogen, and some of them have multiple nuclei, and they are present in the substance or the material of the cardiac muscle fibers, surrounded by also a connective tissue. So it is a section, actually it is in the moderator band, and the arrow points to the Purkinje muscle fibers, while the star points to the ordinary cardiac muscle fibers. Here you should remember carefully what are the differences between the two types of fibers. The Purkinje are larger, the Purkinje here are vacuolated, the Purkinje here have multiple eccentric nuclei. So these are the features evident in the picture. Here we have the last picture in this trial exam. It is a picture for a longitudinal section. Of course, we have here cylinders, but here the cylinders are branching, and if you concentrate, we have single central oval nucleus, so it is an LS in the cardiac muscle. While here the cylinders are not branching, and we have multiple peripheral oval nuclei, and this is the connective tissue, of course, of the endomysium with its cells and fibers. So it is a longitudinal section in the skeletal muscle for two reasons. We have the multiple peripheral nuclei. We have no branching of the fibers evident here. We have more evident striations, transverse striations. So it is an LS in the skeletal muscle. Here we have the cardiac muscle, here LS, the cylinders, but they are branching, and we have single central oval nucleus, and we have a very important, this one, it is the intercalated disc. Here, if you see, you can notice that it looks like stairs, زي السلم. We have transverse part, we have vertical part, so these parts here are actually two types of junctions. We have the gap junctions, and we have the desmosomes. So this is the intercalated disc. But the types of junctions are not evident in the light microscope, but you should remember what are the two types of junctions present in this intercalated disc, and what are the functions of this intercalated disc. Of course, these are RBCs in blood capillaries in the endomysium in between the cardiac muscle fibers.